What if I told you that a simple fluid choice costing less than $2 could potentially impact thousands of hospital deaths? Today, we're diving deep into the fluid trial, published in the New England Journal of Medicine in June 2025. This massive study compared lactated ringer solution versus normal saline across entire hospitals, not just ICUs or emergency departments, involving over 43,000 patients. I'm going to break down what this means for your practice, where the study succeeds, and more importantly, where it falls short. Let's get started. Crystalloid fluids are among the most common interventions we use in hospitalized patients, yet the debate over which one is superior has raged for years. The SMART and SALTED trials showed mixed results in ICU and emergency department settings, with some meta-analyses suggesting potential mortality benefits for balanced solutions. But here's the gap. Most evidence comes from critical care settings. What about the thousands of patients on medical wards, in operating rooms, or step-down units? This hospital-wide approach represents a paradigm shift, asking whether entire health systems should change their fluid policies, not just specific departments. Let's quickly review what makes balanced crystalloids different. These are intravenous fluids with electrolyte profiles closer to plasma, lower chloride content, and buffers like lactate or acetate. Think lactated ringers, Plasmalite, or Hartmann's solution. The theoretical advantage? Lower risk of hyperchloremic acidosis compared to normal saline, potentially reducing renal injury. The evidence is compelling in specific populations. Faster resolution of diabetic ketoacidosis, reduced adverse kidney events in emergency departments. But here's the catch. Balanced solutions cost more. When you're talking hospital-wide implementation, that difference adds up quickly. The question becomes, does the clinical benefit justify the cost? The fluid investigators asked a deceptively simple question. Does hospital-wide use of ringers lactate reduce death or readmission within 90 days compared to normal saline? This isn't just about individual patient outcomes. It's about health system policy. Should hospitals stock balanced crystalloids as their default fluid? The implications are enormous when you consider the volume. Over 330,000 liters of fluid were used across just 7 sites during this 24-week study. The design was elegant. A cluster randomized crossover trial where hospitals used one fluid hospital-wide for 12 weeks, then switched to the other after a washout period. Originally planned for 16 hospitals, but COVID-19 forced early termination after only 7 completed both phases. This is our first major red flag. The study lost more than half its planned power. The 43,626 patients came from diverse settings: emergency departments, medical wards, ICUs, operating rooms. It's one of the largest randomized trials comparing these fluids, but that headline number masks a critical issue we'll discuss shortly. Patient characteristics were typical. Average age 59, 58% female, representing a broad spectrum from elective surgery to critical illness. The hospital-wide implementation was comprehensive. At least 80% of crystalloid inventory had to be the assigned fluid, with automatic substitution orders to ensure compliance. But here's where implementation reality hit. Adherence to lactated ringers was only 78%, compared to 94% for normal saline. This isn't just a statistical footnote. It reflects real-world barriers to balanced crystalloid adoption that hospitals face every day. Staff familiarity, supply chain issues, and prescribing habits all matter when you're trying to change an entire institution's practice. The primary outcome was a composite of death or readmission at 90 days. Secondary outcomes included death, readmission, emergency department visits, initiation of dialysis, total hospital length of stay, and discharge disposition to a facility that was not the patient's own home. The primary outcome tells the story: 20.3% composite death or readmission with lactated ringers versus 21.4% with normal saline. The adjusted difference was -0.53 percentage points with a confidence interval crossing 0, statistically insignificant at p-value of 0.35. Every secondary outcome followed the same pattern: no significant differences in death, readmission, dialysis initiation, or hospital length of stay. The forest plot of subgroup analyses shows consistent null findings across age groups, surgical types, and comorbidity levels. No serious adverse events were reported with either fluid. But here's what's interesting. Nearly every point estimate favored lactated ringers, despite not reaching significance. This pattern suggests we might be looking at a type 2 error. The study lacked power to detect a real but small difference. Credit where it's due, this trial has remarkable strengths. The crossover design minimizes confounding by using each hospital as its own control. The pragmatic approach, with over 40,000 patients from both academic and community centers, provides excellent external validity. The comprehensive hospital-wide intervention reduces contamination bias seen in previous trials where patients received different fluids in different departments. The use of validated administrative databases with over 99% accuracy for mortality and readmission eliminates missing data concerns. And the safety profile is reassuring. No serious adverse events reported with either fluid. Now for the critical analysis. First, the elephant in the room. COVID-19 forced early termination, reducing power from 16 to 7 hospitals. This isn't just about sample size. Statistical power isn't linear, and losing more than half your planned sites can dramatically reduce your ability to detect clinically meaningful differences. Second, the adherence problem I mentioned earlier reflects real implementation challenges. Lower compliance with lactated ringers may have attenuated any treatment effect. Third, relying on administrative data means we lose clinical granularity. We don't know individual patient fluid exposure or have detailed physiological data. The broad population is both a strength and weakness. While it enhances generalizability, it may dilute effects in high-risk subgroups where balanced crystalloids show clear benefits, like sepsis or diabetic ketoacidosis. So what does this mean for practice? The study suggests both fluids are safe for general hospital use, but doesn't provide compelling evidence to mandate system-wide changes. However, absence of evidence isn't evidence of absence, especially when underpowered. Cost-effectiveness analysis, notably absent from this paper, is crucial for health system decisions. Even if balanced crystalloids offer small benefits, the higher cost may not be justified for universal use. But condition-specific evidence still favors balanced solutions for diabetic ketoacidosis, sepsis, and high-risk surgical patients. The implementation challenges highlighted here: staff education, supply chain management, adherence monitoring, are real barriers that health systems must address regardless of which fluid they choose. Here's my bottom line. The fluid trial represents an ambitious attempt to answer a health system level question, but COVID-19 disruption left it underpowered for definitive conclusions. The consistent point estimates favoring lactated ringers, despite non-significance, suggest we might be missing a small but clinically important benefit at the population level. This raises two critical questions for discussion. First, should hospitals change their fluid policies based on this trial? I'd argue no, not based on this study alone. The evidence isn't compelling enough to overcome the implementation barriers and costs. Second, how do we reconcile this with condition-specific evidence favoring balanced crystalloids? Here's where personalized medicine meets population health. Perhaps the future isn't universal policies, but targeted use. Balanced crystalloids for high-risk populations like sepsis and diabetic ketoacidosis, while normal saline is used for routine maintenance. The fluid trial reminds us that even well-designed studies can be derailed by external factors. As we navigate evidence-based medicine in an uncertain world, we need to be comfortable with nuanced conclusions rather than black-and-white answers. What's your take? Are you convinced by the null results? Or do you think we're missing something important? Drop your thoughts in the comments. I read every single one. And if this deep dive into trial methodology was helpful, smash that subscribe button for more evidence-based medicine content. Until next time, keep questioning the evidence.